Hi, welcome all. Welcome to our channel Microbiology Made Easy. So in this video, I'm going to discuss about the B.Sc Nursing Microbiology important questions for the university exam. So here I'm giving the expected university questions. For the university questions, they may give the according to this, they may ask the questions. So here it is, important questions are the first from part A. So which has classified two parts, part A and part B. Part B has as having the infections control and biosafety, it will be there. So here I am giving the part A applied microbiology important questions. So the first chapter is the general microbiology. Under general microbiology, we are having Robert Koch postulates and phenomenon. Next, contributions of Louis Pasteur. So they may ask about parts of optical microscopy, shapes and arrangement of bacteria, whether it is cocci, bacilli, tetrads, arsenic will be there. So the arrangement you have to write. So gram staining they may ask. So here I am giving the details of 4 marks and 10 marks question. So I will explain you which is 10 marks, which is 4 marks. So I will stress for 10 marks. The remaining will be the 4 marks. So here it is. The next one is AFB staining. The next structure of bacteria, bacteriological cell wall. That is, they may ask difference between gram positive and gram negative cell wall or structure of bacteria. Long questions they may ask. Write in detail about the structure of bacteria. You have to write capsule, cell wall, cytoplasmic membrane inside the internal organs which is present. So everything you have to write. The next is structure of flagella. The next arrangement, monotrichous, locotrichous, amphitrichous, it will be there. You have to draw the diagram. The next is about the bacterial growth curve, lag, log, stationary phase. Next, types of culture media. For 2 marks, they may ask enriched media, enrichment media, transport media, aerobic culture media. Then selective media, they may ask. The next is about aerobic culture methods. Under that, most important is the McIntosh field jar method. This is important. The next is collection and transport and processing of specimens. So they may ask, how will you collect the specimen, that is urine sample, pus sample, sputum sample, and then transport and processing. The next important is for long note, they may ask, what is infection? Write about the types of infection, primary infection, secondary infection, iatrogenic infection, different types will be there. You can go through that. Next, difference between exon and endotoxin, that is for 4 marks, they may ask. The next branch is immunology. So under immunology, we are having important questions on immunity and its types. They may ask for long notes or for short notes also. They may ask HAP10, incomplete antigen is known as HAP10 for short notes. The next is antibody and its types. So what is antibody? Right, IgG, IgM, IgD, IgE, IgA. They may ask individually. They may ask IgG, IgM. Next explanation about the antigen antibody reaction, that is agglutination, precipitation types you have to mention. The next is ELISA. What is ELISA and the types of ELISA? Indirect ELISA, direct ELISA, complete ELISA will be there. The next is what is hypersensitivity and its types. So what is, you have to define hypersensitivity and types. Type 1, type 2, type 3, type 4, them you have to mention. Next, for individual 4 marks, they may ask type 1 hypersensitivity and type 4. For long notes from immunology, they may ask antigen antibody reactions. The next is hypersensitivity reactions, they may ask. The next is bacteriology, bacteriological infections, that is pathogenic bacteria. They may ask staphylococcal infections, streptococcal classification. So pneumonia causing bacteria, pneumonia, they may ask gonococcal gonorrhea, meningococcal meningitis, diphtheria causing bacteria, DPT diphtheria, pertussis tetanus vaccine, they may ask anthrax bacilli. The next is gas gangrene, that is Clostridium perfringens, food poisoning, that is Clostridium botulism, tetanus, Clostridium tetani, they may ask UTI causing bacteria, that is urinary tract infection, that is caused by Escherichia coli. The next is dysentery, bacillary dysentery, that is Shigella, that is enteric fever, typhoid fever caused by Salmonella. You have to mention pathogenesis. For long notes, they may ask cholera, Vibrio cholera, bone infections causing bacteria, that is Pseudomonas. You have to write TB, tubercle bacilli, mycobacterium tuberculosis. Next, leprosy, mycobacterium leprae, they may ask. So important for long notes is they may ask typhoid fever. Most commonly, they may ask typhoid fever and cholera. So remaining for short notes, we can expect that. The next is we are having the important chapters. The third one is the pathogenic viruses. So pathogenic viruses are morphology of virus, they may ask. Next, herpes infection, varicella zoster infection, polio virus. They may ask about the polio pathogenesis, they may ask, and prophylaxis. Next, Influenza virus, measles, mumps, rubella, MMR, they may ask. Rabies virus, prophylaxis also they may ask. Dengue virus, hepatitis B virus pathogenesis and lab diagnosis. Same for HIV also. For HIV, hepatitis are long questions. Remaining will be, except rabies, remaining will be the short notes, 4 marks. So you have to concentrate on these. The next is important fungi. Fungi are superficial mycosis, dermatophytes. Next, mother of food disease, dimorphic fungi, two morphological forms fungi that is named as dimorphic fungi. Candidiasis, most important, cryptococcal infections. Next, opportunistic fungal infections, that is Aspergillus penicillosis, they may write. So this is about the mycology. The next is parasitic infections. So parasitic infections are, everyone knows that, important life cycle pathogens of amoebic dysentery, most important. Next, giardiasis, giardia lamnia, trichomoniasis, trichomonas vaginalis, Kala Azar disease. Next, Plasmodium, most important. Plasmodium classification and pathogenesis lab diagnosis of malaria. So malaria and the amoebic dysentery are long questions. Remaining will be for short notes. Next, difference between Taenia solium and Taenia saginata. For short note, they may ask. Tapeworm, that is Echinococcus, dog tapeworm. Sorry, this is dog tapeworm. Echinococcus granulosus. The next is parasitic infection under this. Life cycle pathogenesis of roundworm, Ascaris. Next, short notes, hookworm, Enterobius formicularis. Next, NIH swab. NIH swab for short note, that is National Institute of Health, for 2 marks. Next, pathogenesis lab diagnosis of filarial worm. So these are the important questions. You can go through this. You will get good marks for the university exam, that is for NTR UHS and the KNR UHS. So these are the most expected questions. So I have gone through the different papers. So here they have given according to the new syllabus. I was prepared with this. These are the important questions. So thank you. Please do subscribe my channel, Microbiology Made Easy. All the best for your exams. So thank you very much.